So in addition to being able to see all of your wonderful faces and visit with people from all over the world in this virtual setting, one of the other great things about a virtual conference is that if you happen to come down with incredible tooth pain and have to have emergency oral surgery and a root canal at the exact same morning of your talk, you can pre-record your talk and send it to your wonderful moderator, Dr. Linda Rayer, and she will play it for you. So hopefully I'm with you this morning, and hopefully my surgery went well. But I am going to record this talk and tell you about Amblypygids. So I'm going to change the title of my talk to actually just Amazing Amblypygids, because this is going to very much be a walk through some of the super cool research that my lab and Dr. Vern Bigman and Dan Wigman's lab at Bowling Green State University have done over the past several years. Okay, so we all know what Amblypygids are because we're at the Arachnological Congress. These are arachnids that have 8 legs, but they only walk on 6. They have these really cool spined pedipalps. And one of their most unique features is that they have these incredibly elongate antenniform legs. And when you look at the tip of these antenniform legs, they're covered with hairs. And amazing things they can do is that they can smell. So Amblypygids can detect airborne odor. When we go in and take a look at the brain of Amblypygids, we see that the processing center for olfactory stimuli is huge. And that led myself, Dr. Wolfie Gronenberg, Dr. Vern Bigman, and Dr. Dan Wigman to put together an NSF grant several years ago now to look at the neural basis of complex behavior in these incredible creatures. So Sky Long, Dr. Sky Long was the first postdoc on the project, and this is a 3D rendition that she created of an Amblypygid brain. And I want you to note the 2 different purple colors are the lobes of the mushroom body. The green are the calyces. The blue on the sides are the optic lobes, and then the kind of red at the bottom is the arcuate body. And what you should be able to see is that these mushroom bodies are enormous. They are absolutely huge. Now, what we don't know is what the function of these mushroom bodies is. In other arthropods, mushroom bodies are thought to be sensory integration centers and thought to be used for learning and memory. So one of our first questions was, what inputs go into these amazing mushroom bodies of Amblypygids? And this is work that came out of Wolfie Gronenberg's lab along with Sky Long in a recent paper. And what you can see here is the blue is olfactory inputs and the magenta are visual inputs. You can see in the calyces of the mushroom bodies, they're receiving inputs from both olfactory and visual neurons. And it looks like the olfactory neurons are going into these large glomeruli and the visual neurons into the smaller glomeruli. This is just kind of a cartoon rendition. On the left side is the visual projections going into the mushroom body, and on the right side, the olfactory projections are going into the mushroom bodies. Okay, so the mushroom bodies are multimodal. So how are Amblypygids using these antenniform legs and the mushroom bodies, and how are they using olfaction? So some earlier work that I presented at past meetings involved work in the field where through some various sensory manipulations, we were able to show that Amblypygids navigate using olfaction. So this is using displacement studies where we manipulate their sensory system, displace them from a tree, and look at whether or not they can get home. And we also did some radio telemetry. We want to look at this more closely in the laboratory. And so one of the first questions that Patrick Casto, who's a PhD student in Dan and Vern's lab at Bowling Green State University, asked was whether or not self-derived chemical cues help Amblypygids identify a refuge. So he designed an experiment where individuals were introduced in an occupied shelter, and they were provided either a shelter that had their own cues, the self-cue shelter, or shelters that were either blank or had a conspecific cue in it. And when you look at the difference in occupancy time between these two alternatives, being the self-derived cue or the blank, and the self-derived cue and the conspecific, you see that Amblypygids spent more time in the self-derived shelter. So this red line indicates equal spending of time in each of them. And you can see that they spend more time in the self-derived shelter than in the blank. So they're likely using olfactory cues during these last stages of homing when they're identifying and choosing a shelter. Okay, can they learn olfactory cues associated with a refuge? So we know that they use them, but how are they acquiring them, and can they actually learn them? So again, this is a study out of Bowling Green State University where they designed a box where an amblypygid started, and there were 2 shelters associated with it. In each of these side shelters, there was a plastic screen that the amblypygid could not get through. There were water-laden sponges, so humidity was equal in both shelters. And then odors were placed in each of these shelters. An amblypygid started in the start box. There was a barrier. It was lifted. It could wander into the center and try to get into a shelter. One of the shelters was always blocked, but the other shelter was always open. So there was a negative and a positive stimulus. In order to look specifically at the effect of olfaction, they created 2 different treatment groups. So a sensory derived and a sensory intact group. For sensory derived, they clipped the tip of the antenniform legs because this is where the olfactory sensilla are. So here, if we look at the x-axis, we have sensory intact, sensory deprived, and the association index is how long they spent in the positive, in the trained cue, basically. And for sensory intact, it was between 80 and 100% of the time. For sensory deprived, it was much lower. So it looks like these olfactory cues are what they're learning, and they need the tips of their antenniform legs to do this. Okay, they also wanted to ask if they have good memories. And so 2 weeks later, they ran a retention trial. And when we look at that, we see that the retention trial is just as good as the sensory intact initial trials. So they can remember, and this fits with previous work that I've done with Dr. Casey Fowler-Finn, where we showed that up to 3 weeks later, opponents could remember each other after agonistic interactions. Okay, so one of the next questions we wanted to ask is if they could actually exhibit multi-configural learning. So are these multisensory inputs, visual and olfactory, or other sensory inputs processed independently, or do they actually form a configural representation? So to ask this, what you would do is train individuals to a multimodal set of cues, and then test them with each of them independently. If they're processed independently, you would expect that they would perform just as well with individual cues as they would the multimodal one. However, if they're integrated in a configural representation, when you train them on a multimodal stimulus, you would not expect them to be able to respond well to individual stimuli. An important note about this design is that previous work had shown that amblypygids could learn tactile cues, specifically sandpaper. So this study involved odor and sandpaper as the 2 multimodal stimuli. On the top left here, you see individuals were exposed to either odor or sandpaper for the single cues, and then they were paired exposed to all those combinations of odor and sandpaper in the multisensory cue. Overnight, they were trained to associate one of those cues with an open shelter, and then they were tested with closed shelters to see how long they stayed close to the shelter that they were trained on, to the cues that they were trained on. What they found is that when they are only exposed to olfaction and texture and trained to olfaction and texture, they do really well. More than 60% of the time, they stayed on the side with the trained cue. So they're learning. In the multimodal training one, when you separated out olfaction and texture, they did quite poorly, but they performed very well on the multimodal test. So this suggests that they are learning the configuration of olfactory and tactile, and that when you break that apart, they no longer recognize it. Okay, so amblypygids can use olfactory perception in the field to find their homes. They can use self-derived odors to choose a refuge. They can learn olfactory cues associated with the refuge. They can remember olfactory cues associated with the refuge, and they can engage in configural learning. And all of this is in part responsible, their mushroom bodies and olfaction are in part responsible for this. Okay, so the very last thing I want to tell you about is a study where we tried to ask if they could learn concepts. So could they learn something like same? So there's a lot of literature in the Hymenopterus, specifically in bumblebees, showing that if a bee learns that when it enters, it goes through a yellow hole, then to exit, it needs to match that. So this is called a match to sample test. So it needs to find the yellow in order to get out. And bees can do this with visual patterns, with colors. They can even do it with multimodal stimuli. So we wanted to test this with amblypygids. We used odors, we used geraniol and 1-hexanol. Individuals were with odor in the starting chamber. They went through a 20-minute training trial. They were trained 2 times a day for 5 days. Then they were tested, they were retrained, and then they were tested with a novel odor. What we expected was that if they can learn this concept of sameness, the trained amblypygid should choose the matched side in both novel and non-novel tests, and they should spend more time in the matched side in both novel and non-novel tests. What we found is that in novel tests, there was no difference, but amblypygids initially chose a matched side more often with a novel stimulus, suggesting that they are learning this concept of sameness or matching. There was, however, no difference in the proportion of time spent on the matched side in test trials. Okay, so amblypygids can do all of these things, and in addition, they can potentially learn concepts such as sameness. They are absolutely amazing. I love working with them, but they are also incredibly challenging to work with in the laboratory. But we have some other really cool experiments going on right now that I hope to be able to tell you about in person next time. So with that, there are lots of people I would like to thank. I would like to acknowledge the lands that some of this work was done on. And I'm guessing that other talks have been running late. I'm not sure if I'm going to be able to answer questions or not. I'm not sure if I can talk, but if I'm here, I am happy to answer questions in the chat. Thank you so much for your patience, and that's it. Thanks.